Right. So I'm sure everybody can see my screen, and the topic is not as controversial as it seems. Some of the ideas which we've been exploring over time. I did my PhD in Cambridge in John Carr's lab, and afterwards we've been able to have a long-standing collaboration with him, working on a couple of viruses for crop protection, but mostly understanding plant virus vector interactions and how we can exploit that for crop protection. So I'll just get into it, and this probably is true in most cases that viruses have been attracting very bad press for quite some time, and at least in the African context where I've done quite a bit of my work, we've had quite recent problems. We had maize leaf necrosis a couple of years ago, ravaging the entire maize growing sector in East Africa. And this was a combination of two viruses, maize sclerotic motor virus and a series of other potiviruses, which synergistically just caused 100% losses. But maize is not the only staple that is affected by viral diseases. We've got cassava viruses, which are a constant threat to West Africa, where cassava is the main threat. And so a lot of the work which we've done then, myself and colleagues, has been to understand how viruses manipulate this interaction. And the key question at the back of all this has been, if we make an adjustment in one direction or the other, will we make things better or worse? And what's on the screen is just a snapshot of what is now basically accepted, that when a plant is infected with a pathogen, and in this case a virus, and most viruses are transmitted by aphids, so that's why hence the aphid picture. Then there are changes to the volatile blends, the size of the plant and color, which are perceptible by aphids. And this could alter the orientation of the aphid and cause it to choose to land on that plant. But we also know from the behavior of aphids that when they get onto the plant, they'll start by probing. And there they do encounter a whole series of different cues, which then determine whether they settle on the plant for long-term feeding, or they will disperse without feeding. And this behavior basically is what causes transmission to continue, but it's a lot more complex depending on the virus, the plant, and the aphid species. But at least for the Carr lab, which I joined in 2013, done a whole body of work which I couldn't possibly do justice to speaking about it by myself at this moment. We do know for work which we've done in Arabidopsis and in tobacco, and with cucumber mosaic virus as the host virus, that CMV infection causes the plants to become distasteful. And it then deters the aphids from long-term settling. And this is how the cycle of transmission then continues. And when I came into the lab in 2013, there was now the effort to move this whole body of knowledge into a crop of importance. And for East Africa, it was the common bean. Now, common bean in East Africa is a very big deal because it's the main source of protein nutrition for majority of the people there. But it's also grown commercially by smallholder farmers on plots of land between 1 and 5 acres. Now, its cultivation is affected by two very well-known potiviruses, bean common mosaic virus and bean common mosaic necrosis virus. But there's also quite a bit of cucumber mosaic virus over there, for which there currently exists no means of breeding for resistance. Now, while there's significant work which has been done with bean common mosaic virus in breeding for resistance for the I gene, in East Africa, it is the bean common mosaic necrosis virus, which induces systemic necrosis, that is endemic. And so we've got an excess in futility here, because where the I gene is not, which is a dominant gene, is not protected by the recessive BC3 gene, then you get this trailing necrosis, which then kills 100% of the plants in the field. Now, also more interesting is that recently we've been seeing the occurrence of recombinants and resultant viruses, which are able to break well-established forms of resistance. And this could, with global trade, it's just a matter of time before this gets into the places which is required most. So we also know that two aphid species, Aphids fabae, which is a bean specialist, and Myzus persicki have been identified as the main aphids within these farms and associated with the transmission of these viruses. And so then most of the work has then rotated around these two aphids and these two potiviruses and cucumber mosaic virus. But maybe just to step back a little bit and to explain a bit on the transmission. So these three viruses are transmitted non-persistently, and non-persistent transmission, as you already know, is characterized by the very rapid acquisition of the virus as the aphid probes the epidermis. And again, the transmission is equally quick. As it probes on a new plant, then it loses the viral particles pretty fast. And so if you look at it in the context of being in a field, then it's very easy for the viruses to spread from one plant to another. And so we sought to then understand this three-way interaction within common bean, because we now knew that it's not a one-size-fits-all. And depending on the host plant, the reactions could be different. So that we may know that if we make certain interventions, we'll be making things better or worse. And I'll try to summarize this work done over quite a couple of years and went on even after for the PhD on what we're able to find out and really conclude. And so what we really found out was that there are changes to the plant, or at least to the cues that the aphid can perceive while on the plant, which affect the time with which it begins to probe. And so what we found out was that aphids begin to probe faster on virus-infected plants. However, we did notice that phloem feeding is affected by virus infection, especially for CMV, which is quite understudied. We noticed that the E1 waveform, which is associated with the aphids encountering difficulties feeding on a plant, were enhanced when the plant was infected, and there was generally lower time spent on phloem feeding when the plant was infected, which meant that the plant was not a really suitable host when infected. And this was further confirmed by work which we did with choice experiments, and we saw that in the push-pull thinking, for bean it's more of a push, and the aphids prefer plants which are not infected, not that they'll not land once they're infected, but over 24 hours, if you observe them, you'll find that more aphids will settle on an infected plant. And if you look at it in the context of virus transmission, then this is how the virus will move from one plant to another. We're also able to deduce that there were changes to the volatile profile, and we were able to distill down to specific volatiles where we saw specific changes. For example, we had alpha-copain, which at least in the fruit fly research is a potent attractant, was depressed or much lower on virus-affected plants. There were changes to levels of methyl salicylate, which for some aphid species is considered as a signal for a favorable host. But these changes were also quite virus specific. But we were more keen on the entire blend, because one thing which we learned from our colleagues at Rothamsted, who are collaborators on this project and in their previous work, was that aphids are more perceptive of the whole blend rather than individual volatiles. And for this here, not really clear, you can see like all the virus infected blends are in green and purple and pink, and for the more inoculated plants is here in blue. Changes do occur, which then are perceptible by the aphids. So what do we do with this kind of information? And the next slide is really work done by John and collaborators, both within Cambridge and in East Africa. And there was this attempt now using this whole body of work brought together to try and disrupt the dynamics of the transmission of potiviruses using mixtures of beans. And there was work which went in to look at whether specific bean varieties were more attractive and others were not, and to try different configurations to see whether we could disrupt virus transmissions. I think this is a plot of land whereby all these different mixtures have been done, and John's lab really is a custodian of this data. I've just borrowed it. But this is an example of how now we were able to move the work on plant virus vector interactions into real-world studies to try and affect what the smallholder farmers would do. But we didn't stop there, and we then pivoted and moved our work into high throughput sequencing. And these were things really got interesting, at least for me, in the context of what you could do with viruses. And in these two pieces of work, we then discovered the presence of insect-infecting viruses. And for what we found in bean farms and maize farms in Kenya was aphid paralysis virus and Big Sioux River virus. Another time when we were able to detect these viruses, there were no complete genomes for BSRV, for example. Others were the first one. And we also found Rhopalosifam paddy virus. Now, these are insect-infecting viruses. They do not cause any pathology within the plants. They do not persist in the plants. So if you track them within a plant by RT-PCR, after they've been inoculated by either an insect, you find that within 2 weeks they clear. But what is known is that they utilize the plant vasculature to access parts of the plant. And when aphids then land on the plant and they feed, they pick up these viruses into their body. And we also know that they do cause varying levels of pathology to aphids. So for example, aphid paralysis virus is known that for Myzus persicae, it will outrightly kill that aphid species. But for some other species of aphid, it could cause disorientation or just having these different levels. So we thought, oh, this is something really good we want to study. But we also did more metagenomic approaches. And in another case with collaborators in East Africa, at Becker, we found Andona viruses. Now, these are persistent viruses, which again, don't cause any pathology in the plants. They're inheritable and they're not transmitted. It's not possible to mechanically inoculate a plant with Andona virus. And at least for some studies, they have been associated with being mutualists, which may confer certain properties to the plant, including increasing their resilience to insect herbivory. So then what my colleagues and myself began then to develop was a high throughput sequencing driven approach to protecting crops. And what we were really keen on was to study the diversity of insect-infected viruses and the percent viruses for starters, and then downstream to investigate the potential in limiting the transmission of harmful pathogens by aphids. But in this picture, what I really want you to appreciate is the kind of landscapes that you work in. And these are areas which in Kenya we were able to collect samples from until we got to where we were. But if you look at the bottom left, there's a lady here, and this is her farm. So this is in Nakuru in Kenya, for those who've been able to travel there. And she's got 5 acres of land. This is a mixed maize and bean plot, but where she's standing, she grows forage grass for her cows right next to the bean farm. And so quite complex ecosystems. And at the top here, this was almost like a monoculture maize farm, although right next was beans next to it, but this farm did not have stuff mixed into it. But at the bottom here, on the bottom right, we've got maize, beans, fruit trees, brassicas, kale, and everything. And these are the kind of farms which we find. And so when we looked at the work which we were doing and what we wanted to achieve, we decided then to target the maize-bean intercropping system to sample for the diversity of these cystoviruses. And we did find stuff. So we now know that at least for majority of the sites which we sampled in Kenya, you will find the Big Sioux River virus in common bean and in the bean specialist Apis fabae. You will find aphid paralysis virus in maize, common bean, and in Apis fabae. And we found this in the maize aphid Rhopalosifam mydes. We found the Hubei 55 picona like virus. So now we do have at least 3 candidate viruses which we can begin to interrogate for their properties. And we did start this work of in collaboration with the group in Cambridge with aphid paralysis virus. So this is what we are actually investigating in terms of the value of these viruses. And the part in pink is what we're used to. Bad outcomes, for example, CMV, causing this attraction to aphids and repulsion. We've got this transmission of viruses. But what we hypothesized was, if we introduce the cystoviruses, then we can break this death cycle by either causing the killing of the aphids, or causing disorientation, which would make them more susceptible to predators. And we're also keen to investigate on the role of pollinators and other insects in the dispersing of these cystoviruses, because in literature we had found that some of these viruses get shed through the fecal matter of these insects. And so by using high-throughput sequencing, we were looking whether there's a common virome between these different players. So this is work which we'd be keen to continue with. And we did make some progress. Unfortunately, it's now in abeyance due to the cuts in ODA funding. We hope to be able to pick it up and continue. But that was not all. We also investigated persistent viruses, and this is ongoing work, again in collaboration with John's lab at Cambridge. And we screened 17 varieties of bean grown in Kenya, Uganda, and Rwanda, where we know that beans are a big deal. And in at least 10 of these varieties, we found the presence of at least one endovirus. And one variety, in fact, had got all the 3 endoviruses present. And we've been very keen to get on the whole genome information, again, through high-throughput sequencing. And for Phaseolus vulgaris endovirus 3, this is quite good because this is the first time it's been reported or found in varieties grown in East Africa. And currently we do have a master student who is investigating some of the properties of these viruses in varieties of bean which harbor this. And hopefully when we come back later, maybe we should be able to give more evidence. But in summary, what I can say over the 9 or so years of work that we've been working on these different aspects is that we now know that understanding plant virus interactions is critical in developing crop protection methods, and especially for IPM strategies. Now, not all of them will work, but they're good clues in there what can work well. We now know through high-throughput sequencing that there are other virus players that can affect insect transmission or alter host plant qualities. And we are testing and working out on the premise that these are good viruses, and they are viruses which can be used for as new avenues for crop protection. And I'd like to thank all these people. I probably have missed a few who have been supportive during this work across different areas and collaborations across Africa, within Kenya and Ghana. The Royal Society, which funded my work through a fellowship, ICP, where I was, and of course my colleagues at Cambridge, and all these people who have contributed time and money to make all this possible. So thank you. I'll now take your questions.
Thank you, Francis. A very quick question before I open to more expert people. How long has this research going on, and how much is known about this?
So the in-depth research into the cystoviruses and the endonaviruses began in 2019. So I was fortunate to win one of those Royal Society Flair Fellowship, and I set up my work at ICP. And so we had got 2 years of dedicated work just working on this, and we did win an additional grant to pursue for another 3 years. Unfortunately, Corona happened, and the cuts in ODA funding mean that the samples are stuck in -80 freezers.
Okay, so it is quite recent.
Yes, yeah. It's quite recent, yeah.
Wow, okay, that's amazing. Who would like to open the discussion, and we can even pick up the earlier discussion if anyone would like. Please, questions, comments for Francis, for the group.
Oh, Nick.
Hi, Francis. Nice to see you again.
Nice to see you.
You didn't say much about how successful the cystoviruses can be in terms of killing the aphids or disorientating them, what sort of effect they can have. I wonder, is that well known?
At least, maybe in only one case, I think there's been an investigation on ALPV on Mysis persicae. But as I mentioned, because we now know that, at least from what other people have done, that it's quite species specific, we were then keen on testing this on the aphids which we had. So together with John and Alex, we did get a full-length clone going for that, but disruptions the past 2 years then.
Okay, I say I probably raised horrible memories of what you or horrible issue weren't able to do because of the pandemic. I'm really sorry about that.
Yeah, we should have had the answer to that question by now.
May I ask one other question?
Yes, please.
You mentioned the role of pollinators in moving them about. So I guess you introduced this virus into a field, one of these viruses into a field, or you might try that as a crop protection strategy. How then do you ensure that it moves from plant to plant?
So our initial take on this was we wanted to know whether it happens naturally. So we had got a bit of the project that was presented, which concentrated on pollinators. And so getting pollinator samples and then trying to get the virome of that in fields where we had already detected these cystoviruses and seeing whether they could detect them within the bees.
And so, but your contention is that they're already there, so we don't need to move them around.
Because we were a bit worried that unless they were there and the bees were doing well, then if we introduced them, we're not going to cause any harm happening. But if we found it, then we could say that this is already occurring in nature. And then we could now rig up a system whereby we could be able to use them as living drones, as it were, to spread this.
So thank you.
Martin.
Well, I just wanted to pick up a point on that last one about pollinators. You mentioned the farmer with her 5-acre plot, and she was obviously very geared up to crop diversity, but I think her particular crops weren't ones that would attract a lot of insects. And I wondered whether there were a range of insect pollinated crops that might come into that story within that region.
Yeah, so indeed they were, but hers was a very interesting farm because by the time when we got there, she had sprayed. So it was an extremely clean farm, with the exception of the areas which were not sprayed, so like the trees, and there was a potato patch, and there were plenty of pollinators going there. One of the things which we noticed about that, we thought we'd find plenty of honeybees, but intriguingly, within that landscape, we found more of carpenter bees. Again, these are largely understudied, and so we've ended up trying to get more into the carpenter bee space, trying to understand whether they the key ones, and they are fascinating to observe, honestly, because they behave a bit like bumblebees. They're very systematic in how they pollinate from plant to plant and all that. Unfortunately, our attempts at high throughput sequencing were not that successful. It's something which we'd be keen to repeat to see whether we can be able to capture the viral effect.
Thank you.
Kostya, please.
Thank you. Francis, nice talk. The endoRNA viruses that you mentioned in the end on beans. So you discovered them through sequencing, high throughput sequencing. So do you already know whether they actually affect interaction with insects or not? The presence of those viruses? That's the first question. And the second one, whether they affect yield, for example, in the plant, so whether their presence is associated with yield penalties or not, or they're completely benign.
Yeah, I mean, it's a good question, but that's part of the work which one of the master students which we have at Cambridge is beginning to tease apart in terms of we are more keen on the insect side, not yield at the moment. We want to see whether the presence of these endoviruses will affect, will increase or does confer some resilience to the crops against insect herbivore. I don't know if I can throw John into the deep end of this to release the data.
Yeah, sure. Well, with the endoviruses, the effects on insects, I don't think are well studied. But with the what used to be called cryptic viruses, another group which just get transmitted in the same way without apparently causing disease, there is evidence from a group run by Marilyn Rusink that they repel aphids. So that's one of the things that's well, we were already interested in these things before that paper came out. She has some unpublished data apparently, which suggests that those pepper plants because this isn't pepper, it's different crop, but the pollinators are more attracted to them. I mean, pepper isn't utterly dependent on bee-mediated pollination, but it is enhanced, so that might be a useful thing. There's a couple of papers came out recently which have slightly rained on our parade. I think there's two main groups of these endogenous viruses: the endornoviruses, the endogenous RNA viruses Francis mentioned. Another group, the used to be called the cryptic viruses, the partiti viruses, and then another one called the amalgo viruses or something, which is just like doesn't matter about these names, but there's a couple of papers come out on the amalgo viruses, suggest actually that they actually are deleterious for their host with respect to if a different virus comes along and infects them, it increases the amount of the infectious virus. So, it may not always be a good thing, but for the value of helping farmers, if you can say, because let's say when people breed plants, they track DNA, they don't track RNA. That gets thrown away down the sink. So if you could say, well, have a look out for this, and if it's this type of inherited RNA virus in this particular crop, then maybe you want to make sure that's not going to be in your germplasm anymore. For the following reasons, and it's just as valuable for breeders to be able to look out for a bad trait as a good one. So, but insofar as these other things like physiology and growth and yield, there's little packets of information around, but nothing comprehensive, which is really what Francis, I think, was or is aiming to achieve.
And the idea is actually to employ those viruses as kind of biocontrol agents. But the idea is to identify the volatiles and then deploy volatiles in the field to either repel or attract, repel aphids or attract the.
It might be. It's such an early stage.
Okay.
We thought by now, Francis, didn't we, that we'd be chock-a-block with information. We got this 2 years to work on it. And COVID at a year and a half of it. We got lucky to get some sort of extension really.
But it's fascinating. Yeah, thank you.
But Francis, have you been doing these experiments here in the UK or in Africa? Or both?
So, most of these experiments were done back in Kenya. There was also a significant collaboration with John, again funded by the Royal Society, which has enabled this to go on. So yeah, we are hoping that
they've been done.
pick it up again.
Yes, I can imagine. Anyone else? Any comments, questions? Can't see if there's anyone raising hands. No? Don't be shy if you do. Francis, would you like to add anything? I mean, I wanted to ask you if this research can be applied to other regions, of course, not just Kenya and other regions in Africa, but also say Asia, and in a way it's the same way of working going to apply and the same outputs?
Yes, it can. We've had quite a nice collaboration with a team at the University of Ghana who've been working with mostly cabbage, and we did some deep sequencing for them, and we did detect a couple of diacestoviruses there as well. So my own thinking is that these viruses would probably be ubiquitous in nature, but we still have to be very meticulous in studying what they do, what we think they do, and then that would possibly give them broad application across different cropping systems and across different places in Africa. For the endornoviruses, I think this probably could have a much more global application due to the possibility of them being used in breeding or being bred out, depending on what we find. And that's work which can even continue here in the UK, and maybe some of the valuable crops which
yeah. Oh, okay. Teresa, would you like to ask a question, make a comment, please?
Hey, Francis, nice talk. Have you thought about how to deliver the endonoviruses to aphids or plants?
Oh, those ones are a bit tricky. I think if we were able to do that, would be extremely rich.
Mm. Yeah.
But what the plan really, when we began this work, was to see that if we could prove that they do confer certain, advantages, could we work with well-established breeding programs and see whether this would be part of the selection process. That went in that way, then it would be very, very valuable indeed, because it's something which nobody would consider in terms of the moment. And so that maybe that would be a new way of adding value to breeding programs. We are in contact, at least within Africa, with Cabra and Ecabren, who are the large networks around bean systems within. We hope to be able to work with them downstream once we have proved what these things possibly do. Then,
yeah. Thank you, Teresa. And Francis, you talked about intercropping. I mean, now this is probably an obvious question, but could flowers being intercropped with the crops be an additional but beneficial? Beneficial addition.
Yes. So
no difference observed.
Yeah, like having wildflower patches within. Yeah, I think so too. I think that would be good, or at least for carpenter bees. What we noticed is there's a complete loss of like habitat for them, and they're treated as a bit of a nuisance. And maybe that's because nobody really knows their value as pollinators. So they'll typically drill holes within poles or in your house, and then what a farmer normally does is to plug those holes because they don't see the value. And then this habitat loss for them. But maybe if we're able to prove a beneficial role that these things can offer you some sort of ecosystem services, then there will be the motivation to have those wildflower patches, but also within the fences or whatever place where they are, then they can maintain areas which would be sustaining to carpenter bees and all that. But it's still early days. It's still early days.
Yeah. The intercropping, I guess, one resistance or one point of resistance could be then it becomes more difficult to harvest, I imagine, because then unless you could have the clearly defined areas, then I guess it's a bit more difficult, right? And that could be a resistance. Anyone else before Francis can maybe wrap up and give us a sort of a last message? Any more questions or comments? Francis, would you like to just say a couple of ending words before we say goodbye and wrap up?
Yeah, I mean, thank you for attending. And I think as I mentioned, these areas where we have got good preliminary data, we do have a plan as to what we'd like to do. And I'll be pretty happy to, if anybody wanted to collaborate or share ideas around how we can advance this work, I'll be more than happy. Yeah.
Do you know, Francis, sorry, just a quick question. Do you know Lynn Dix in zoology? Because she works a lot on pollinators. I wonder if you don't know her yet, she might be a good person to talk to. So I'd be very happy to introduce you.
Yes, that's
lost Francis.
Sorry.
We've lost Francis, but
it's my camera.
I think you're still there. It's fine. So I'll put you in touch with Lynn. I think it might be good for furthering your conversation.
Yeah.
Right. Oh, you're back. Excellent. Wonderful. If nobody else then would like to ask a very final question or make a further comment, then I would like to thank Francis to join us today. And yes, and I hope that you will join us also next week. We have Daniel actually who joined us today to talk about pulses and healthy diets and sustainability, etc. So it will be wonderful. Thank you, Daniel. So I'm looking forward to that too. So thank you so much, and have a good weekend, and hopefully see as many of you as possible next week.
Yeah, thank you.
Thank you so much. Thank you, Francis.
Bye.
Take care.